So in this simulation, I'll be using gel electrophoresis to separate three different dyes. And these dyes different size, with the yellow dye being the smallest, the blue dye being the larger, and the purple dye being the largest in that order. These dyes were all equal in negative charge. How far do you predict the dyes would move in their lanes during gel electrophoresis? So taking into account that they're all equal and negative charge, we would separate them based on their size. The yellow being the smallest will travel further because we've read that in the context. This other one would be the blue dye in the second and the purple in the third. We're assuming these based on their sizes because we're being told that they're equal in negative charge. So read to the protocol, prepare the electrophoresis box, centrifuge the solution tubes, drop one solution one, pipette solution one into well one. So I'm gonna follow the protocol. Select the agarose gel. Selected it. Place it into the electrophoresis box. Orient the wells so towards the negative electrode. All right. Pour the flask with one sodium buffer over the gel box. Okay. Select the microcentrifuge to open the lid. Please move all your samples over the centrifuge. Select and move each of the centrifuge to the microcentrifuge. Okay, I'm gonna do that. Balance the microcentrifuge so fill receptacle symmetrical. Okay, I'm gonna check their balance. Balance and ready. I'm gonna close the microcentrifuge. Press the pulse button to briefly spin. Select the lid. Remove the tubes and put them back into the rack. So I close it. Make a note in your notebook to show which solution is pipetted into each lane. Okay. Now I'm going to note that I'm writing down. I'm gonna select the P20 micropipette, this one. Okay. Select the volume and change it to 10 microliters. Let's open the P20 box. Select the S1 solution to open it and place the micropipette tube. Drop one solution. Always close the lids to prevent cross-contamination. Yeah. Select the P20 micropipette and move it to the gel to prepare. Move the micropipette to lane 1. Dispense solution by pressing the plunger to reach its first stop. All right. Move the pipette and eject the tip. Okay. So now pipette solution 2 into lane 2 and lane 3. So we're gonna do the same thing. So we're gonna take solution 2 into lane 2. Close it. Put it here. Lane 2. Done. Gonna select another tip. Close it. Solution 3. put the micropipette back. Place the cover over your electrophoresis box. Select the red and black cables and connect them. Turn on the power supply. Set the voltage to 130. Set the timer to 10 minutes. Okay, start the timer. Wait until time has elapsed. Turn off. Unplug the electrodes. Yes, I'm trying to remove it. All right. Carefully remove the cover and inspect your gel. Look at the relative position of the dye bands in your gel. The bands in your gel have different colors because our samplers are color dyes. Typically, the bands in the gel wouldn't be colored. In the ideal result of this experiment, the blue band would have migrated the least during the gel, the purple band would migrate farther, and the yellow band the farthest, yes, because of the size. Compare results with the ideal results by making a note and describing any differences you observe between the results. Explain why the bands ended up in these relative positions. Is this what you predicted will happen? So as we can see, these are my predicted results, and these are the actual results. We can see that the yellow band traveled the farthest, and here the purple band traveled the second farthest, and the blue traveled the least. And these are the ideal results. The ideal results are similar to the actual results. So I can see here that the yellow band moved the furthest in my prediction, as well as the actual and ideal results. However, the blue and purple bands differed because here in the actual experiment, negative charge affects them. Bands of the DNA. So that's why the purple band is different from the blue band, since the purple band I can't write anymore. The purple band, so that's why the purple band is has more of a negative charge. Negative charge. Because in the predictions, they told us the negative charge was equal for all of them. But here in the actual experiment, we can see that here what matters is a negative charge, and we can see that purple is more negative charge than the blue. That's why it traveled furthest. That's because in the actual experiment, we deal with the negative charge and the size compared to the predicted results, which we were told it was only the size of the band. Based on size, you may have expected the blue band to migrate further than the purple dye. Yeah, we expected that. In practice, the purple band, this is because of the fine, yeah, the charge determines. What part of the procedure do you think had the greatest impact on the results you obtained? I think it was the proper handling. the solutions and pipetting into the wells, else the results would have been affected or erroneous. The part I think had the greatest impact on the results. So the reflection. Let's look at what you learned from the simulation. Please click on your answer each of the following questions. Study your electrophoresis results. Which sample contain a single dye and what evidence for your experiment supports this choice? This would be the S3 bond based on the number of bonds. Yes. What charge did the dyes have in this experiment? They were all negative because they moved toward the positive pole. The dyes separating in this experiment simulation are orange, G, yellow, phenol, bromo, phenol, blue, purple, and xylene, cyan, blue. These dyes were fragments of DNA. Which color will represent the smallest piece of DNA? It would be yellow because that's the one that traveled the furthest in the experiment. If you compare two molecules that were found in the blue band in the gel, what could you infer about them? Do they have the same size and charge? If you run a gel, like the picture below, where would you expect to see a band in the gel? I'd say A, because the positive molecules will travel to the negative electrode in this. As you can see, all questions correct. And I feel very confident. Thank you. And that's it for this lab. I will read the summary on my own.